Okay, let's go on to my next guest. Joining me now for a look at some of the latest on the Zika virus situation, related research, etc., is Dr. Vincent Racaniello. Dr. Racaniello is a virologist and a professor of microbiology and immunology at Columbia University. Dr. Racaniello, welcome back to the show, my friend.
Hello, Robert. How have you been?
I'm not too bad. I see you've been all over the place.
I stay busy.
Yeah, I see that. You're hard to keep track of, but you got a lot of information, good information going on, and I want to touch on that a little bit later in the interview. But let's talk about your blog, Zika Diaries. It's been about 3 months since we last talked about this, and what you do on this, it's a blog on the American Society for Microbiology, and it kind of chronicles your lab's work on Zika virus, which is new to you guys.
That's right. We worked on polio for over 30 years, and with that about to be eradicated, we've been looking for other viruses to work on, and Zika virus we picked up early this year. And I thought that I would use the blog Zika Diaries to document a kind of a personal view of our travel through Zika, and we'll see how long it goes.
Right. Now, I went back to September, and I found this pretty interesting piece. You did a grant application for support from the March of Dimes.
Right.
And I thought the whole narrative there was pretty interesting. Can you talk about that story a little bit?
Right. So the March of Dimes was actually founded by Franklin Roosevelt in the 30s, I believe. He wanted to raise money to combat polio, which he had himself, of course, and which was paralyzing a lot of people in the U.S. and elsewhere. So he did fundraising through the March of Dimes. He raised $500 million, which was a lot back then.
Sure.
And that money went to developing the 2 polio vaccines that we use today, and which have almost eradicated the disease. Now, once the vaccines came out in the 50s and 60s, and it became clear that they were going to control the paralytic disease, the March of Dimes stepped back away from polio, and they started to work on birth defects. They actually worked on rubella virus for a while. They funded work on rubella virus, which causes birth defects. And now, with the emergence of Zika virus and its cause of birth defects, the March of Dimes is supporting work on Zika virus. When I came to Columbia in 1982, I could not apply for March of Dimes funding because I was working on polio. And I just think it's ironic that now, 34 years later, I can finally apply for March of Dimes funding. So we sent in a grant application for our work on Zika. And we're going to hear later this month, later in November, to see how we fared with that.
Oh, good. Good. I was going to ask you about that because I didn't see you talk about that on the blog.
I will. Rest assured. I'll let everyone know.
Well, I mean, and this kind of leads into the next question is the most recent post you did is you filed an application with the NIH for a grant. Mm-hmm.
So just before the March of Dimes application, we filed a different one with the NIH, a 2-year application for a rather small, they're meant to be exploratory grants. They're called R21s, and that's to do work on Zika virus. And that was reviewed just 2 weeks ago, and unfortunately our score that we got is not good enough for us to get funded, so we're not going to get money for that application. I don't know yet what the reviewers did not like. I'm waiting for the summary of that, and hopefully we can address some of their issues and send it back again.
Gotcha. Since August, I think that's the last time I had you on the show. What has your lab been doing?
So I have a rather small lab. I have 3 people. I have 3 young ladies who are working really hard, and most of them are working on Zika virus. We've got a lot of different isolates from all over the world, about 15 or 20 different isolates that we've collected from Africa to Asia, South America, the U.S. We're making DNA copies of them so that we can modify the genome, because we'd like to know if there are any biological differences between these isolates, or are they all the same. We've also been we made a model of studying Zika infection in mouse embryos. So we found that the virus infects mouse embryos, especially the brain, very well, and it has effects on brain development, which probably mimic what's going on in microcephaly. And so we're trying to push that forward. We need funding because mouse experiments are pretty expensive. So hopefully we can get some. Otherwise, it's not going to go on very long. And the last thing we do, you might be familiar with this virus, enterovirus 68, which
yes, I am.
Popped up a number of years ago, and now there's some anecdotal reports that there's more paralysis in young children, which may be related to this virus. So we actually have some funding to work on that virus, so we're continuing that. We want to know, is the virus neurovirulent, and if so, why?
Yeah, well, actually, that's been a big piece of the news in recent days, because the CDC just put out new numbers, and the cases really skyrocketed in the past month or so. So, well, considering they went from like about 50 to 90 or something like that. So
it's funny, we kind of pushed that work to the side with Zika, but we're starting to refocus on it because I think that's going to be important for a while. But
it really has it been solidly confirmed that that's the cause? I mean, it could be a number of viruses, right?
Yeah, it's really hard because there's so many viruses that are infecting these kids. I mean, we know that it causes respiratory infections, common colds or maybe even more serious, but whether it's paralyzing the kids, we just don't know. We don't even know if the virus has the ability to grow in the central nervous system. So that's one of the things we're trying to figure out with our mouse model.
Gotcha. All right, let's switch gears and talk about some other stuff that's going on in the world of Zika. As if we haven't seen enough medical issues related to Zika, they seem to be popping up all the time. I saw a study from researchers at Washington University School of Medicine in St. Louis, and it looks like, based on what they did, it may also affect the male reproductive system. Can you talk about this study at all?
I sure can, because the PI on that study, Michael Diamond, was just here yesterday in my office, and I recorded an episode of our podcast.
Oh, good.
So I heard all about this. So basically, as you know, Zika virus can be transmitted sexually. It's not as frequent as mosquito transmitted, but it is. It's present in semen for long periods of time. So what these individuals at WashU did is to take mice and infect them, male mice, infect them with Zika virus, and see what happens. And it turns out the virus replicates in quite a few places in the mice, but one of the highest levels of virus is produced in the testes. And the virus gets in, it replicates for, the length of their experiment was 3 weeks. In humans we know that it's there for at least 90 days or so. And in the mouse, it destroys the structure of the testes. So after 21 days, the sperm that are produced, and the virus infects sperm as well, they're aberrant, they're not able to fertilize females very well. The testis goes from the normal size to about 10% of that size. It's hugely shrunk. The tissue structure of the testis is destroyed, and the mice do not recover from that. So this is all in mice, but the results are really convincing. And this raises the question: should we be looking in males to see if there's an effect in this way as well?
Well, no doubt. I mean, when I first read that, I was like, are you kidding me?
It's amazing.
Yeah.
Every week it seems there's something new associated with this virus.
Sure.
And I should say dengue does not do this. They use dengue as a control in this paper.
Oh, okay.
It does not replicate in the testis of male mice.
So, I mean, who would have thunk this, a few years ago?
Absolutely
what's
not.
Zika virus? Now, this is not really directly virology related, but it's close enough. It looks like that Colombia and Brazil are going to be launching a Wolbachia program. And this is to battle their immense mosquito-borne virus issues down there. Can you talk about what this is and how it works so my audience could understand it?
Sure. So Wolbachia is a bacteria, and these are bacteria that have to live inside another cell. They can't exist on their own. Wolbachia, and they infect about 40% of the world's insects. Many mosquitoes have Wolbachia, but it turns out Aedes aegypti, which vector dengue and Zika, among other viruses, does not have a natural Wolbachia. So in the lab, people have found if you put Wolbachia into Aedes aegypti, it makes them resistant to viruses, including Zika and dengue. And we don't really understand how it does that, but it seems amazing. And so the idea has arisen, let's take Aedes aegypti and other mosquitoes, even Aedes albopictus, which is another vector for some viruses. Let's put Wolbachia into them and then release them into the wild and try and populate the world with Wolbachia-containing mosquitoes, and maybe it'll reduce virus transmission. So a few release studies have been done where they take these Wolbachia-containing mosquitoes, release them, and then say, do they survive? And in some studies, the mosquitoes survive, they propagate. And so we don't know if that has affected virus transmission yet, but now they're doing several bigger releases. I understand China is releasing millions and millions of Wolbachia-containing mosquitoes. Brazil and other South American countries are going to be doing this. We've gotten past the point where people are worried about releasing a genetically modified organism, because that's what this is, to now, will this decrease virus transmission? And I think it's a really exciting way to look at it. There's no pesticides involved, and it could work.
Oh, that's great news. And I think there still is a small anti-Wolbachia crowd out there.
I'm sure.
Yeah, but I think that's shrinking. Now, I got about 4 minutes left, so I'm running out of time. But I wanted to get your thoughts on this. This is the first time I even heard of this story, and it's probably been going on for a while, about Zika testing controversy at the CDC. Do you know anything about this, Dr. Racaniello?
Well, I've been looking into this at your suggestion, and this is really an interesting story. And it has to do with a PCR-based test for Zika virus. One of the best ways we can diagnose Zika virus infection now is by looking for virus nucleic acid using the lab test polymerase chain reaction. And a long time ago, Robert Lanciati, who works in Fort Collins for the CDC, he developed a Zika PCR test during the outbreak on Yap Island because he wanted to figure out what was going on there. It was a test designed just to look for Zika virus, and that's been used up to the present. Now, remember, no Zika diagnostic has yet been FDA approved, right, but you can use them under emergency conditions. Now, recently the CDC decided that they want to be able to analyze specimens from patients, not just for Zika, but for dengue and chikungunya. So they developed a triplex assay, PCR assay, to detect all three. And they're recommending that this be used. But Lanciati feels that this misses a lot of positive Zika infections, which are detected by his test. So CDC denies that this is the case, and he and Lanciati have been locked in a discussion about this. This has been going on since the spring. And in fact, back in the spring when he first complained about the triple test, CDC actually demoted him in his position. And then he complained, and they restored his position because that wasn't appropriate. So CDC says you're wrong, and Lanciati says CDC is wrong. It's gotten to the point where the U.S. Office of the Special Counsel, that's the lawyer for the president basically, had to write the president a letter by law telling him what's going on here. So I don't know what the outcome is going to be. I know Lanciati has been working on these viruses for 20 years or more, since the 80s, and I think he knows what he's doing, and I think the CDC needs to work with him and sort this out.
Yeah, I can't believe that I missed this story for the bulk of this year. It's not something that the press has been on, really. Yeah. All right, well, let's switch gears. I got about 2 minutes left, and I want to go ahead and talk about what's new on Virology blog, virology.ws. Now, you're not only you have your little media empire over there, and so you got the blog, and you're doing podcasts all over the place. So what's new, Dr. Racaniello? Well, I just finished writing a story about a new Ebola virus study that came out of 2 laboratories, and what they did was they found a mutation that arose in the Ebola outbreak in West Africa, the recent outbreak, seems to improve infectivity of the virus for human cells and decrease in infectivity for bat cells. So the idea is this mutation arose early in the outbreak. It dominated the entire outbreak, has never been seen before, and the authors of the study think that it might have something to do with, human infections. We don't know whether that's the case or not, but it's being picked up all over by the press this afternoon, and so I thought I would put in my 2 cents on that. Yeah, and also apparently your podcasts are doing pretty well. I mean, mine goes on Stitcher, as does at least one of yours, maybe all of them. And you're in the top 100 in the science section. Well, I think that's great. I love it that people want to listen to all stories about infectious diseases. Yeah. Yeah, you're doing a great job. I love it. I love listening to them. I just don't have all the time to listen to all of them. You got too many of them.
Well, I got news for you. There are going to be more. So.
I have no doubt. You're doing a great job. All right. And the website, again, is virology.ws. Thank you, Dr. Vincent Racaniello, for your time and expertise once again, sir.
My pleasure. See you in a couple of months.
Yes, sir.
Bye-bye.
Have a great holiday. All right.